Thank you very much for that introduction and hi everybody. It's a pleasure to be here today. I wish it could be in person. It's been a very interesting year. We're doing a lot of our work online, including fisheries, which is quite a difficult thing to teach online. Really, it's all about getting out there, and so hopefully this year's better. But you know, hearing some of the research in the medical area gives me great hope. Australia really leads the world in that sense. But I'm not a medical researcher. I'm a marine ecologist, a marine fish ecologist, as a matter of fact. And so I want to talk to you a little bit today about some of our work and some of the very basic tools that we use. So I'll switch over to my slides, if I can do that. Here we go. And feel free to put things in the chat line, and I'm looking forward to discussing whatever you want to afterwards as well. So there I am up in the Southern Barrier Reef collecting data, and it's very old school. We jump in the water, we use a slate, an underwater writing slate, and we look at things. We look at the corals, we look at the fish, we look at their behaviors and that sort of thing. And so I will say at the outset, a lot of our work involves observation of nature. We work in the lab as well, but there's all sorts of issues with taking fish away from nature. So there are very big questions out there, of course. You'll all know about climate change, overfishing in the oceans and things like that. And all these big problems, to do some science behind this, to underpin what we respond to these issues, involves some pretty basic science. We need to know about fish in the case of my area. How old do they get? Where do they move to? And then how can we protect them? A marine park's a good idea. Are we really overfishing? If we're managing fish, are they staying in one area? Are they moving? Some fish move across oceans. How do we know these things? So I'm going to talk a little bit about some of the tools that we use. So a fish lecture would not be complete without a dissection or a cross section of a fish, would it? And so this particular fish, this bony fish, I won't go through the anatomy, but up on the top left, you'll see a little thing called an otolith. Otolith literally means ear stone, and it's found inside the fish's head. And you might ask, so what? Well, it turns out this ear bone is really the equivalent of a diary to a fish. So if we catch a fish, it's hard to go back in time. We haven't yet taught them to speak or understood their language. So we rely on indirect means. So this otolith is a pretty amazing thing. It's a little bone. It sits in the inner ear. And as the fish moves, it has a bit of inertia, so it helps the fish with balance. But it's also useful to us because it grows in a concentric manner. And so there's a picture. This is a very tiny, maybe 10, 15 microns, so a bit thicker than a human hair. Once we cross-section this little stone and polish it down, we see these lovely rings. Now, these concentric growth rings. So in the middle are the rings put down by the fish in the ear when it's a little baby. And then in this case, each day, it puts down a new layer of tissue, and that tissue is mainly calcium. And so it puts down this tissue each day. So obviously, we can count these rings, much like the rings of a tree, and work out how old the animal is. And that's pretty fundamental knowledge if you're trying to work out the ecology of species, their age distribution. Now, in humans, we do things called censuses, where people come around and ask you questions in your household, how many people live there and how old they are. So we can get that information that way, but we can't do that with fish, and so we rely on these ear stones. Here's another one. That's actually an ear stone of a very large fish, and the rings there are put down every year. They're called annual rings, and you can see this particular fish has been dated, so each of those dots represents a year back in time. So there are rings put down by that fish in 1960, etc. So fish get very old, and in fact, one of the oldest vertebrates ever caught is this rockfish. And it looks old, doesn't it? It looks very ancient. And the otolith that goes with it is on the left. And it was aged over 200 years old. So this fish was around well before Charles Darwin wrote Origin of the Species, for instance. The Industrial Revolution, etc. It hung in there until the poor thing got caught off the coast of America. So the otolith is very useful for that. It also has a number of other uses to us, apart from the use for the fish. We can age it, as I've already said. We can also measure the width of the rings And that gives us an estimate of the previous growth of the fish. Again, much like a tree, there's a whole science called dendrology where they do this to trees. You can look at growth rate and you can see summer growth and winter growth, and you can do that with fish as well. And another much more recent thing we do with the otoliths is, see those little holes there? I'll talk more about them in a minute, but they're basically what they call ablations, small holes where tissue is removed, and we can analyze the chemistry at each point. So given that we know the middle one is when it was born and the outer rings are just recently in terms of the fish, we can look at how the chemistry changes, and I'll tell you what that can do in a minute. So I'm going to talk about just a couple of quick projects that quick information on big projects we do and how the otolith gets involved. So you'll recognize this. This is not a photograph I took the other day. It's of course, the movie Nemo, and we are the Nemo people. We've been studying the tropical fish coming down the coast from Queensland for about a bit on 20 years. In fact, we just did some surveys the other day, and they were the start of our 20th year of counting fish off the coast of Sydney and further south, and the Barrier Reef. So in Nemo, the fish arrived down the East Australian Current into Sydney Harbour, and that's exactly what happens. And Dory there, the blue one, we've actually found one of these in Sydney Harbour, but Nemo not yet, just for your information. So there some of them are. That's a blue damselfish and a Moorish idol, looking a little bit lost perhaps, but they really do like these rocky habitats. They don't like the kelp as much, they like the rocky urchin habitats. And our very simple model of what's going on is shown here, where we have the distribution of the fish on the Barrier Reef, with the red being their breeding area and the outer yellow being where the babies come, because the way tropical fish move, or any fish just about, is they release babies, and it's the babies that do the moving. In this case, they crank on down the East Australian Current and actually end up shifting southwards, which is what's happening, and the babies go further south as well. And that's basically what we're investigating, what's happening, and is it a function of climate change, which I won't get to today. But this is not the Barrier Reef. This is off the coast of beautiful Sydney. So you'll see a couple of different species of tropical fish there. And where do they go to? They like these coral habitats. And this is new coral habitat that's arrived in Sydney in the past few years. It's absolutely stunning, sitting on the bedrock. And again, through the years, we've seen the numbers of fish, numbers of species here and further south increasing. So back to the otolith. So here's a cross-section under environmental scanning electron microscopy, showing these rings that I spoke of earlier. And we've been able to determine that the wide rings in the middle are when the fish is in the ocean and it's a baby, it's hatched up north and it's come down. And the yellow portion there represents, in this particular case, when the animals arrived at Sydney, found a place to live, and that's what's happened since. And so, as I mentioned earlier, we can count the rings. So we can work out, for instance, we could ask the question, how long has this fish been at large in the ocean before it got to here? We can also measure growth rate. So as an example, we've done some work with these little fish. This is a little butterfly fish, a baby butterfly fish that's been found not just in Sydney, but further south near the Victorian border, a place called Merimbula. And we're able to work out that this animal was 60 days in the ocean based on the otolith counts. And by using oceanographic modelling and reversing it to find out where the currents would have been, we've been able to backtrack, done thousands of simulations. If the fish landed at this red spot, where would it have been 60 days beforehand? And we've been able to plot distributions of the source of these fish, which is very important for us to know where they're coming from. So the otolith has been very, very helpful in that. Sorry about the slightly complicated graph here, but this is the coast of Australia, East Australia, as you know, all the way from the north. Lizard Island is on the northern part of the reef, all the way down to Merimbula, down south. And the little cartoon just shows the seasonal movement of the East Australian Current every summer. It brings warm water and these babies down with it. And so by looking at lots of these otoliths of several species of butterfly fish, we looked at how long they'd been in the plankton. And as you can see, Lizard Island there, about 40 days. And down, but interestingly, way down at Merimbula, all the way down south, also 40 days. We'd expected much longer. The only much greater time at large has been Lord Howe Island, which is that beautiful island shown on the right that we work in. And that's probably because to get there, it requires a lot of extra travel on the East Australian Current. So the otolith has told us a lot about the travel of these animals so far. Moving rapidly to another study we do, we work on another creature called the freshwater eel. Some of you will be cringing, but these are actually beautiful animals. They don't have spines and they're very, very, very gentle, it turns out, luckily. These are my fisheries colleagues and students. And this particular girl is 60 years old about when we aged her with her otolith. And what we're interested in is the life cycle of these creatures. Some of you may know the eel, freshwater eel, which also occurs in Europe and other places, is a big delicacy, but also has a fascinating life cycle. And this is really just to show you how complicated it is rather than to go through it step by step. But basically the fish moves between fresh and salt water. It gives birth and hatches its young up in deep water in the ocean. And then it moves, the babies move down to a freshwater river where it lives most of its life until it goes up to breed again. And our particular model suggests that different species move from our coast here up onto the Coral Sea. Very, very difficult to get a handle on this. If you tag a fish, you'll never see it again. They're just so rare. And so in comes the otolith again. And here's a cross-section of an otolith of about a 15-year-old animal. And you'll notice these little dots again. And so what we do is we use this machine. And as an ecologist, it's quite exciting to get to the lab and use a fancy machine. This is the LA inductively coupled plasma mass spectrometer. And what it does, it shoots little holes in our otolith where we tell it to, takes a small amount of tissue at that point and analyze it through mass spectrometry to give us the elemental signature, what elements are in there. And you can see those dots, they're about 10 or 15 microns wide, and they represent each about 3 or 4 months of the fish's life when you look at them closely, from birth in the middle to the edge, which was when it was caught at about 15 years. And what the theory is that this calcium, when it's in an environment, the fish is in an environment that's of a certain type, it takes up some of the elements from the local environment. So if it's in salt water, it turns out it takes up more strontium, and it picks up divalent ions, like calcium is a divalent ion, so it'll take up strontium, manganese, iron, and all those sorts of things, lead, all sorts of elements, which are of interest to us. So in other words, it's again a diary of the local environment. So just to give you an example, this particular otolith, and there's its little ablations, I've plotted the strontium concentration at each of those points. So the left-hand side is when it was a baby, had a very, very high strontium content, which is indicative of pure ocean water. But after about a year or so, it dropped dramatically, and the strontium content of that particular hole was very, very low, indicative of fresh water. And then towards the end of its life, it was caught in an estuary, in a salty downstream part of a river. It went up. So in other words, it's a little migration tag of that particular fish, which is unique. We couldn't get it any other way except through the otolith. And just incidentally, the top left there is the similar otolith, but we're now plotting lead concentration. Concerningly, in a lot of our animals, lead and other heavy metals go up alarmingly later in the animal's life. So these animals are sort of a bio information on the local environment through time. And if you find a 60-year animal, you've got 60 years of data right there. So we think that's a useful use of an otolith. We also work on sharks. Who wouldn't? White sharks and other sharks. We do lots of different things with collaboration with CSIRO and fisheries. And of course, again, same questions, where are they going, etc. These, you may know these animals don't have bony skeletons, but they do have cartilaginous skeletons, which we still put down local rings. And this is a beautiful cross-section of a shark vertebra. Here's my student Jack down at the Anstone Nuclear Facility at Lucas Heights, and he's dissecting vertebrae. Collections are kept at the CSIRO of hundreds of white sharks that have been captured through whatever means. And then those vertebrae are polished and put through this secondary iron mass spec, which is held there to do a similar analysis that I spoke about with the eels. It generates some beautiful artwork, I think. I think there's a lovely pictures of different parts of the shark's vertebrae, clearly showing those concentric rings, which he's shown to be age rings. So just a quick example, this particular age ring is of a fish caught north of Newcastle. Yeah, there are lots of white sharks up there. And you'll see this particular one I picked was mercury concentration, Hg, which is a major pollutant you wouldn't normally find in a natural ocean. And so you'll see a little spike on the right side of that yellow curve. And we've determined that that is when the fish swam past the Hunter River in Newcastle, which is known to have be a source of some of these chemicals. So we've still work to do here, but we feel that these shark vertebrae can be very useful indicators of where the fish has been. And this is just one example. And finally, we also work on a number of different sea creatures. One of my very, very favorites is the sea dragon. I don't know how many of you know of this animal, but it's found just off the coast in Sydney. You can jump in off Kurnell, Australia's, where there was a settlement 200 years ago, right within 100 meters of that, are major spots where sea dragons are found. Now, they're not actually dragons, of course, but they sure look like them. And they are members of the Cygnathidae family, which is a group containing seahorses you've heard of. These all have the unique characteristic of the males giving birth in a pouch. And so it turns out they're absolutely beautiful creatures. And we've been studying them for about 15, 20 years for various different reasons. We're also very chuffed to hear that David Attenborough claims several times that the weedy sea dragon is his favorite creature on Earth. So he's, I guess, our patron now. They're found all around southern Australia. They go as far north, if you know the coast, as Port Stephens, a bit north of Sydney. And we're trying to investigate why they don't go further and what climate change has got to do with it. All around southern Australia and Tasmania, and up the other side to Western Australia. So they're quite widespread, but they're fairly rare. And they really need that kelp habitat to find their food and shelter. So it's disappearing. So there's an issue there. So in the early days, of course, one of our main goals is to follow individual fish, which we do with other fish as well. And we want to follow them so we can measure their movement, their reproduction, their growth, their survival, all those sorts of things. So individual recognition is important. So we first started off trying to look at whether the fish could be recognized quickly by its little fins, its appendages. So as you can see here, we had complicated diaries of fins because a lot of fish will have a fin missing. But to our disappointment, when we did control studies, we found the darn things grow back very quickly. So a fin missing is good for a couple of months, but if you come back next year, you may not even notice it. So looking at those big natural markings didn't seem to work. The next thing we've been doing is we do this a lot with fish up on the Barrier Reef, where we do a lot of our work on reef fish. We inject them with a small amount of a silicon-based paint called elastomer. And that particular paint is fluorescent colored. So you can see us injecting there. The animals are very docile and we take great care. They're bony, so it's quite a challenge getting this small pocket of paint in there. It lasts forever, pretty much, as far as we know. And the bottom right, we then use what's called an ultraviolet torch to shine it on the flank of the animal, and then it shows up the tag quite easily. Now this worked really well, but there's an issue with it, of course, in that you have to handle the animal, which we'd rather not do. It's really just for scientists and people with permits, and you have to have the special torch. You can't just cruise along and look at them. So it's hard to involve all the hundreds and thousands of divers around Australia in a project that involves this. But we did get some cool data. Incidentally, there's a picture of a male with its eggs on the outside. So the sea dragons do not have a pouch like the other ones, the seahorses. So it's very convenient to be measuring what they're doing there in terms of numbers of babies and that sort of thing. But very quickly, the panel on the left shows data on the numbers at a bunch of different sites in Australia, in Eastern Australia. What we're finding even more recently is the numbers are dropping alarmingly. So we have great concern for this animal. And it may well be listed on the IUCN Red List fairly soon. And it's not direct fishing, it's more loss of habitat, we believe. And there's sites in Tasmania where it's gone and it used to be. We're also able to measure growth rates, like the bottom left is a growth curve of fish from the bottom is Tasmania and the top is Sydney, showing, so we can measure growth rates by recapturing these animals. And they don't move very far. We've found one only male that had moved across Botany Bay. And that probably was with the storm. Otherwise, they live their whole life within 100 meters. And even the babies don't move far. So as you might guess, we've also found very strong genetic structuring around the coast, as opposed to most fish. So anyway, so tagging using those methods is not perfect. But you've probably already noticed the very ornate patterns on the side of these sea dragons. And here's an example. So like other fish, I have friends working on manta rays, people have done it on white sharks. There's special software that analyzes photographs for fine features. So if these particular fish had a fingerprint, an individual fingerprint, this could be useful. And it turns out they do. You can look at the flank of a fish, even though it makes me dizzy to look at, through computer imaging and a bit of your own guesswork. You can match a photo to another photo as being of the same animal. And so we're doing a lot of this. And the beauty of this is you can use what's called citizen science. Divers all around Australia, many who are much better photographers than myself, are now donating their photographs to us to learn about animals, these animals. So as an example, we had a series of photographs sent to us from Cornell in Sydney, and we were able to recognize a fish. And I didn't call it David, someone else didn't. It's not after me, I don't think. But pretty much we were able to match this fish back to 2010. And at that stage, we had 6 or 7 years of data showing it was the same animal. So the beauty of photographs, you can go backwards in time, which you can't do with the other tagging. So we're now getting a really good idea of how long these guys live as well, which is very important for conservation. And finally, more recently, we've collaborated with other researchers to develop a website called Sea Dragon Search. So if you Google Sea Dragon Search, you might want to look more into it. And basically, it's more of a global study, even though dragons only occur in Australia. There's a colleague at Scripps in the US involved. And this is an amazing platform because it allows amateurs to learn about what we're doing, but also to upload their photographs, hundreds in some cases, into a database. And through machine learning processes that some of you may know about, it's able to rapidly do what we've taken ages to do with our computer software, and that is match the animals. So in real time, you can put your photo in if you become a member, and it'll tell you, oh, that's Murgatroyd from last time, or we've never seen this sea dragon before. So we're really now going to start rapidly getting more information about this creature, which can only help its conservation. So that is about all I had for you in terms of information. So I'm really, really happy to chat, discuss anything that you'd like to discuss. I'll just unshare my screen. I hope there's someone out there.
Thank you, David. That was a really, really interesting presentation. Loved learning about how you use machine learning in particular with the animals of the ocean. So we're opening the floor to questions now. If anyone in the audience has questions for David, please send them through in the Q&A down the bottom. So I think we've got one here from MG1. Do you work on the Great Barrier Reef?
MG1, yes, we certainly do. My original training, or my earlier training, was in Hawaii and the Caribbean on reef fish, and reef fish are amazing. And so we're interested in what's called the early life history, the babies and the juveniles, because that's when a lot of things happen. And so we have 25 years of coral bleaching data from the Southern Reef, which we've matched to fish numbers. And sadly, we were hoping to get up last year to continue our 25 years of data, but COVID interrupted, we weren't allowed to go. And so we're looking forward to potentially going this year. We have a trip planned in a couple of days, and we're keeping our fingers crossed on the Queensland border. But yeah, we've got long-term monitoring sites. We study dams of fish, which include anemone fish you might have heard of. And they're a fascinating group of fish in particular. And we've found some down in Sydney too. So yeah, definitely study the Barrier Reef.
Fantastic. Hopefully you get to get up there. We have another question from MG10. How do you think machine learning has transformed the field in which you work, and by extension, all of the biological sciences?
Oh, that's a big one, and I'm probably not the right person to answer because I'm a newbie in this area. A couple of my colleagues work on wildlife, and they are actually using machine learning to work with law enforcement, even authorities looking at quarantine. And so that they're developing tests to rapidly assess if some illegal wildlife's brought in, to rapidly be able to assign it to where it's from. Is it a captive bred thing, or have they actually taken it from the wild? So I know it's being used quite extensively for that. As I said, I'm a newbie, and my colleagues who are using this and developing the Sea Dragon website are going to teach me. But from what I see, it's the ability to rapidly hone in on a solution, which we couldn't do before. And so that's that's a take that I have. So it will definitely, like all new statistical techniques and methodological techniques, it'll revolutionize. Statistics, for instance, has gone a huge way since I learned way back when, a long time ago. And students are now doing a lot more what we call multivariate statistics, so they can understand how animals and plants work in nature.
Thank you, David. We have another question from Kambala. What advice would you give someone who is hoping to become a marine biologist?
All sorts of advice. I do think, and we and other universities take work experience students. That's not a bad idea. You really need to get in and don't just go through the motions. So we have a marine biology course here, and there's quite a few students, but there are certain students, and not necessarily the most academically gifted ones, but they're the most enthusiastic and interested, and they'll come and help in the field and just go the extra mile. It helps them, keep them motivated, but it also, we can assess how they'll fit. We do a lot of work with things like the Sea Life Aquarium or fisheries or the museum. And so a student, whether they've started university or they're yet to start, can sometimes get work placements, if you like. And so I'd reckon I do all that because, you know, like any profession, it's not quite what you think. If you think you're doing marine biology to get away from mathematics or chemistry or lab work or whatever, you're probably mistaken, all those things. But it's the sort of maths and chemistry and lab work that's exciting to you because you're understanding marine creatures. Many of us dive, but not everybody. A lot of students don't dive. They do a lot of lab work. There's a lab at UTS that studies Antarctic sea ice, algae and things like that. And there's a lot of lab work involved in that. So yeah, so I guess my take is don't be put off by what others say. Who knows what's happening in future, but it's probably on the average side of job availability. There are better and there are worse. But what I will say is the students who've persisted and really got enthusiastic and done the extra mile have all succeeded in getting into the marine biology field.
Well, there you go. Hopefully we have some future marine biologists in the audience. Okay, we have another question from an anonymous attendee. What initially got you interested in researching fish?
Oh, great question. That was an awfully long time ago. Back in the 60s when I was a kid, that's pretty sad, isn't it? I was into entomology. I was a butterfly and beetle catcher. And back then, of course, you caught them and put them in boxes. You wouldn't do that these days. And I had an amazing trip to Papua New Guinea with a friend. I went to James Roos when it was a boys' school. And my friend David, his parents were biologists because my parents had nothing to do with that. So I went on my very first flight and went to Papua New Guinea butterfly hunting. It was amazing. But during that time, they took me for a snorkel on a coral reef. And looking back, it was a pretty degraded reef, but it really got me interested. And so went to Sydney Uni, and when I went on in my undergraduate, marine biology was a big thing then because we were learning more about the ocean, and all of a sudden it became in vogue. So I guess there's a whole cohort of us from those years that went on to become marine biologists. And so I like anything to do with biology, that's for sure, especially wildlife biology. But my little niche, I guess, has become fish, freshwater as well. We did work under the frozen lakes of Canada. That was hard yakka. But yeah, tropical and subtropical like Sydney, that's my speed these days.
Oh, the frozen lakes sound very cold.
Mm-hmm.
Okay, we've got a question from James Roos, high school. What are your thoughts on the effects of climate change on the Great Barrier Reef?
Yeah, what an interesting topic. And it's really nice to actually have data, your own data, and have a bit of an idea what's going on, which we do in our lab. Of course, it's a major political football. And this is the sad thing about with scientists get very frustrated, understandably, when they want to keep it as objective as possible. What is going on? What are the effects? But when you're derailed by people saying, no, the scientists are lying, they're trying to get some money, or, I went up there last week and it was beautiful, so it's a scam. It's a bit hard to take when you know the truth. And the truth of the matter is the Barrier Reef is huge. There's 2,000 or 3,000 reefs all the way from near New Guinea way down to nearly down to Brisbane. So it's just enormous, 3,000 kilometres. So there's no one answer fits all. So on the southern reef, where we've done a lot of our work, there's been big bleaching events where the corals have been nailed, and we've seen big effects, but they've recovered in the following years. Whereas on the northern reef, where there are many more corals and many more fish, that has really been nailed in 2016 and 2017. So corals can recover, because basically they turn white because the little symbiotic algae leave, but they can come back. But a huge proportion of those corals actually died. And some of these are up to 100 years old, dead. So there's a regrowth that happens, but it's like when you chop down a lot of trees in a forest, you get regrowth, but they're weedy species. So the reef changes because of bleaching. Even if it looks to the untrained observer like, oh, there's a lot of corals out there, they'll often be the new branching weedy ones, not the big massive ones. And the species that rely on those massive corals suffer, even though, again, tourists may not notice. So some of the northern reef is recovered, but a lot of it is dead, and it will eventually reseed, but probably not in our lifetime. So it's quite dramatic and quite bad. We're seeing certain international coal companies being led into Australia, and that's a major source of controversy. Obviously, that we make money out of it, but surprisingly not much and not many jobs. But the risk, not only of global climate change being affected, in Australia, we often say exports climate change to the world. Isn't that a nice thing for us to do? But also the physical dredging of harbours up on the Barrier Reef for boats to come in and out of that are bringing gas or coal is quite alarming. So, you know, really we're doing very, very poorly. We used to be the nation to look up to for coral reef management, and now I won't say we're a laughing stock, but it's a bit embarrassing. And yes, climate change is real on the Barrier Reef, sadly.
Yeah, it's very sad, isn't it? So we have another question from Sydney Girls High. What is your opinion on the treatment of fish in commercial aquariums?
That's a very good one. I'll start with how we treat our fish, because we do lab work. Most of it is observational. Some of it involves sacrificing the animals. Sadly, the otoliths have to come out if you're going to do the aging. But we're guided by a fairly strict set of ethical principles. First one is, do you really have to do this in the first place? Which is probably the main one. Secondly, is it going to find useful information that hasn't been found before? So no duplication. Secondly, thirdly, I should say, the husbandry when you catch them, the best way to catch them, taking minimal numbers to get statistical significance, looking after them in the lab, having monitoring sheets and all that sort of thing. I'm on our local ethics committee, and it's quite strict and rigorous. If there is an incident, a fish dies, there's a major investigation. I'm not clear that commercial aquaria have the same level of checks and balances. Just like commercial fishing doesn't have the same level of checks and balances that I do if I go out and catch a few fish or have the students work with me. So I feel there's a lag there, and the overriding thing is it's a business. You've got to leave business alone. But I do have some concerns. There's some excellent ones. We work with some excellent commercial aquaria that source their fish sustainably, we think, as far as we know. But there's a lot of money in the field, and so you get a lot of, especially if you're importing from overseas, a lot of dodgy operators, and they'll source the fish from Indonesia using divers that use Clorox to squirt into the reef to catch the maybe 1 in 10 fish that survives. So, if you're very careful, and in Australia, we have some of these really good sustainable operators, but I don't believe they have to follow the same regulatory things that we do as researchers, which is a concern.
Yeah, it doesn't sound great. That was a great question. Okay, we have another question from an anonymous attendee. What techniques do marine conservationists use to conserve certain creatures such as sea dragons?
Well, a lot of the techniques are out of the realm of science, and that's been a real learning curve for me. One of the techniques is knowledge. Obviously, we can supply some of that with our work, but also getting it out to the public. Now, I personally do quite a bit of media work with people who are very interested in fish in the sea, which is kind of nice. And so, first of all, publicising what's happening out there. For instance, today we have a Nature paper came out with a group of us, where we've looked at this movement of fish down the coast and how it's going to be affected by another impact of climate change, which is called ocean acidification, which can affect sea urchins. So there's You know, so that sort of paper is of global interest, in the media. There's an AM story on it this morning on ABC, etc. So getting our work out there is important to us. We work with what's called NGOs, environmental non-government organisations, like the Nature Conservation Council and people like that. Now, they are amazing, those people. They're very knowledgeable. They work, you know, under, you know, very poor conditions in terms of jobs, and there's a lot of volunteers, and they are really on the front line of conservation. So in New South Wales at the moment, there's always been a big controversy about marine parks. Now, marine parks are areas, hopefully, where no exploitation takes place, and that includes fishing. We only have about 1% in the Sydney region. The whole coast is marine park, and there was a proposal to get that increased to 2.5%. Well, the political flack was horrible. And in the end, the minister just conceded and said, we're not doing it now. So that's to leave 98% of the ocean open for fishing. And various groups thought that wasn't good enough. They wanted 100%. So, you know, our science can only go so far. It requires people who understand the social fabric of society, really, which I'm learning very fast. Because to me, it's very, very obvious. Plain as the nose on your face, we need to have protected areas, just like a national park on land. But for some reason, there's very good lobby groups to government that are able to convince them that, no, we don't need any protection. We're doing just fine. So frustrating, but I enjoy working with these groups because of their dedication.
Thank you for that. I have another question from another anonymous attendee. How much do you travel for work, and where do you most often travel to?
Nowhere, no, this year. We do, yeah, so our study sites are quite varied, and we do a lot of our work around Sydney. You probably don't realise what an amazing ocean habitat we have in Sydney. We have the tropicalisation I mentioned. We have deeper sea. We have these amazing harbour habitats, mangroves. So we have a lot of projects on many different topics with students around Sydney. We also work up and down the coast, because the coast from here north and south is really special, because it's a transitional coast. It's changing because of climate change, but we get the tropics up north of New South Wales and a cold temperate down south. So a lot of our work involves field trips up to various sites. We also work on the barrier reef at a little island called One Tree Island, which is actually run by Sydney Uni, and I've been going there since 1991 and seen amazing changes. Actually, I went there in 1986, I lie. And that involves an expedition, really. You go up to Queensland, you take a boat, various boats and helicopters even, out to the island. You'll sit there for weeks or months doing your work and then come back. So they're the intensive periods, and various bits and pieces in the Caribbean, Hawaii, other places, which obviously we can't get to at the moment, which is a bit sad. The students do field trips in undergraduate to Heron Island. We're due to go there in May, COVID willing, and that's where they learn some of the techniques of marine research.
And another question on the topic of COVID was, how do you think COVID will affect marine life?
Oh, what a fantastic question. Well, direct effects. Well, first of all, it's affecting the research on marine life, and that can then indirectly affect how we manage it, etc. The amount of money available for this work for marine work has dropped hugely, and partly because there's only a finite amount of money, and a lot of it, understandably, is going to COVID research. So that's a bit of a negative side. On the other hand, there's some interesting things that happen. First of all, we're seeing huge drops in overseas travel, plane flights, ship movements, even fishing. And the result on that has been, in some areas, a huge bounce back in marine life, in water quality, in air quality. Climate change has been slowed down, at least temporarily. I'm sure we'll sadly catch up again. And so there's all these direct and indirect effects which are positive and negative. One of the positive things I see is COVID and the excellent researchers that we've seen highlighted has really meant that science has been shown to be a really important part of society. And now we have a different American president who seems to recognise this. And certainly in Australia, we do recognise our medical researchers. I think the benefits to, the positive spin on science is going to help all of us, even those that work in the marine field.
Yeah, hopefully. We have another question from Sydney Girls High. Could individual students be useful for helping analyse genetic data?
Good question. I'm not a geneticist, but I work with geneticists. The Sea Dragon research, for instance, my part-time postdoc person, she leads that project. And she has had students work with her because, first of all, you've got to get samples. So what we do with the sea dragons, we can take a very, very tiny clip from one of their fins, like taking a fingernail, and that gets analysed in the lab. So certainly students can be involved in any part of our work. And in fact, we encourage it. That's why I'm at a university, not at a government institute, because I think that's important. You know, there are restrictions. You've got to be trained and there's got to be indemnity, but that's the same with any line of work. So yes, students can certainly be involved.
Well, there you go, Sydney girls. Something for you to get involved in. And we have one last question from MG37. What would you like to see our government do and the actions for it to take?
This could be a long one. First of all, I suppose you have to understand that the poor old government has a number of conflicting things going on, not even mentioning COVID. They have many masters. They have to look after the economic wealth. They have to look after big business. And, you know, that's a topic that I don't really want to get into about how they do that. They have to look after health, education. Everyone wants to pay less tax, but health and education and important things like that come from the tax base. So getting that balance right is really hard, but I really think we need more of a balance towards the social system. Everything from importing people and looking after them to disadvantaged societies, to Indigenous issues, to, in my case, marine terrestrial. There's some really overwhelming issues in Australia. For only having 25 million people in a country the same size as the US, we've done a really good job in stuffing it up, I'd have to say. We've really cleared it. We've really done a bad job. And so really the terrestrial situation is probably worse than the ocean situation. So I'd like a lot of brave decisions, keeping water for the Murray-Darling system and the Snowies, keeping it in nature and not hiving it off as much to irrigation. If you need that much water, should you be farming those crops in that place? Probably an agricultural revolution, which is actually happening in Australia, where people are planting sensible crops and raising animals that can live in those areas rather than forcing European agriculture onto Australia. Obviously, protecting areas, marine parks are one part. There's a global goal of protecting 30% of the ocean. In Australia, we've got about 6%, a long way to go. Antarctica, that's a really big issue. What an amazing uncharted place. And unfortunately for it, there's huge mineral resources, fisheries resources that all sorts of countries, including Australia, are clamoring. So we really need strong leadership from Australia in so many different areas, and really getting back up on the horse of climate change, really showing we're leaders. It'd be nice to see a government do that in future. And the state governments also have a role to play in alternative energy and local conservation and their agencies, like in my case, fisheries.
Thank you. Well, that concludes all our questions. Yeah, it's very interesting. You'd think that since we're surrounded by water, we should do something to protect it and what lives inside it. So yeah, well, thank you so much for joining us today. Fantastic talk, really interesting. I loved learning personally about the sea dragons as well. Hopefully we have some future marine ecologists amongst our attendees today. So thank you very much.
Thanks very much, and please everyone keep in touch.
Thank you.
Take care.